Thank you.
So the bad news is it's me again. The good news is that some of the preceding talks at Patrick and Daniel and Mark Daly have covered a lot of the relevant background here. So we can, as you say, rattle through pretty quickly, I hope. Shall I do this then? Somebody else do that. Okay, so we've heard already about the ethnicity gap in human genetics research. Anyone who was at American Society of Human Genetics meeting in San Diego last year will have heard all about the many talks describing this, and there are now many papers with nice graphics showing this as well. So I think the IHCC has a great opportunity to help address this underrepresentation of populations, particularly from low and middle income countries in genetic studies, and that if we want to enhance our cohorts genomically, then this is the place to do it. So the overarching aim here is to conduct a very large-scale, say 100,000 participant pilot study and sequencing substantial numbers of participants from multiple cohorts. So we've heard already about the ethnicity gap. On the left, you see the geography gap. This is the paper that said that 72% of all the participants in GWAS are from either the US, the UK, or here in Iceland. And we've already heard as well, on the right, you see the ethnicity gap. This is the figure from one of the papers. Note particularly at the bottom, the other Asian category. So in South Asians, which is a population I'll propose to do some sequencing in, we only have 2%. And just next to that is African, 2%, Hispanic or Latin American, 1%. There's a big gap between what's happened so far in European ancestry participants, for want of a better term, and other ethnic groups. And as we've also nicely heard, this has a multiple, what's the problem we're trying to tackle here? It's fine, it's very inequitable, but why does that matter? Well, things like polygenic risk scores, we know they do much worse in different ethnicities. So you see in the left plot here that South Asians and Africans, the polygenic risk scores that have been derived so far perform much worse. Of course, that's not just a problem for countries in Asia or Africa, but of course, in clinics around the world, as polygenic risk scores move closer to the clinic, how do we handle participants of different ethnic backgrounds? So there's inequity even in Western and high-income countries. Rather than just some of the problems, I think some of the value, and we've heard about this to some extent already, is the value. I think Daniel MacArthur described it as each population is like a new drawer of variants that might come out that tell us about new genes. Here's a by no means comprehensive catalog of pivotal variants that were identified that told us about the mechanism, the function of particular genes, and the relevance to disease from sequencing or in some cases genotyping diverse populations. Some of them bottlenecks, Icelanders, Greenlanders, for example, others just by going beyond traditional European continental ancestry. There's value, I think, in going to more and more ethnic groups because one will find variants that disrupt different genes to what we'd find if we just stick with the what we've had so far. So as you see in the slide, the text at the bottom there, really widespread genetic sequencing, and of course the phenotyping to go with it should help to reveal many more of these gifts of nature, if you like. We need to think about what one would do in IHCC in the context of what's going on globally. So this is a visual of some of the nationally funded genomics projects going on around the world. And I suspect we'll hear from you and Bernie tomorrow morning about his latest projections on the numbers of millions of people who will be sequenced around the world over the coming years. But things to note from this slide, these are high-income countries predominantly. They do have a spread of ethnicity and geography. So you see East Asia is covered, for example. We have in Australia, we have some in the Middle East, but we're still not touching many of the poorest countries, and we're not touching many swathes of ethnic groups with these kind of projects. Also worth noting that these projects are often in patients, so there's a sequencing to understand causes of clinical conditions, and or mainly still in European ancestry participants. What else is going on? Well, of course, there are many sequences being generated in well-phenotyped participants. So, for example, industry-funded projects like the whole exome sequencing that's going on in all 500,000 people in UK Biobank, or hundreds of thousands, say, in the Geisinger system. But again, largely in high-income countries, largely in European ancestry participants. Of course, people have been thinking about the ethnicity gap. We're not the first to think about it, and there are other projects looking at sequencing more diverse populations, but they often have different angles. So, for example, trying to build a human genome reference that is more representative of more diverse ethnicities, or looking, for example, like the Centers for Common Disease Genetics here in the US that's trying to sequence somewhat smaller sets of ethnicity-enriched disease sets. I think the unique selling point we have here in the International 100,000 Cohorts Consortium is having many large disease-agnostic cohorts. And I'd like to come back to this paper. This is one of Mark's papers from a few years ago now, but the idea was, how should we design rare variant association studies? We've done our GWAS studies. If we're now thinking about moving to sequencing and finding the mass of very rare variants in populations, how should one do it? And I'll just highlight here the summary from the abstract, i.e. a well-powered rare variant association study needs discovery sets with at least 25,000 cases. So if we want to understand the rare variants that contribute to common complex diseases, we need very large studies. So initial objectives from the proposed pilot study. One could go with the what I see as the scientifically optimal but somewhat expensive option. One could do whole genome sequencing of 100,000 participants for understudied communities from low and middle income countries, and one I'll propose is to study is Bangladesh. Or alternatively, one could think about trying to, as done nicely, for example, in FinGen, as we heard earlier today, to do some sequencing to derive population-specific genotyping chips, which would be much cheaper, and can then, with imputation, one can capture some of the same variation. Why do I suggest Bangladesh as one of the options? Well, this is some details of the CAPABLE program, which is a joint Cambridge-Bangladesh partnership about capacity building in South Asia. South Asians, as we've seen, are largely underrepresented in genetic studies, but very large in number, as we heard earlier, so comprise a large proportion of the international community, not only in Asia, but across the world. It's a low middle income country without a national sequencing initiative. Importantly, it has consent for genetic assays, whether that be locally or internationally, and again, that might be a challenge for some low middle income countries. Daniel MacArthur touched on earlier the idea that enrichment for things like consanguinity might help. So the Bangladeshi population is somewhat enriched compared to European ancestry outbred populations for intermarriage, and therefore more likely to find homozygous loss of function variants. And there are other features listed at the bottom that may make it particularly attractive, which we can discuss. Now, one of the questions, of course, if we want to enrich genetically, as I said, is, do you want the expensive option or the cheaper option? I've just listed here, with increasing information content from left to right and increasing costs from left to right, the kinds of things one can do. And it really depends the kind of question one can ask. If one's interested in doing GWAS of various diseases or traits that we would have in a disease-agnostic cohort with phenotype information, then one could use imputed array data. And similarly for polygenic scores, as we just heard, one might just mine chip and array data. If one's also interested, though, in finding these particular ethnic-specific variants that are enriched in a particular population or that one wouldn't find in other populations, then one probably needs to think beyond just array data and beyond imputation, think about sequencing. One could think about whole exome sequencing, which would give us maybe the coding variants, which might be the most immediately tractable and interesting. However, I think I've posted at the bottom there some of the summary from last year's meeting, which I think thought that the niche for this consortium, or the interest maybe in thinking about a more sustainable, more long-term vision and going for genome sequencing rather than just exomes. Of course, all of the projects within this consortium will need multiple cohorts. So one question that I think is pertinent for the breakout session tomorrow is to think about what other cohorts should we do? Should we think about other parts of South Asia and really target one ancestral group and really try and address that ethnicity gap? Maybe Africa, maybe South America, both, or other parts of the world that are clearly deficient so far in amounts of genetic data. Or one could think about more innovative designs, for example, looking at Bangladeshis and other countries, so trying to fix the genomic content, but look at different environmental or lifestyle backgrounds with the same genetic content. Clearly, there are many challenges here. For example, how might one maximally benefit the studied communities? So how do we work very closely with the local community that would be studied to make sure that they are well informed, well involved in the research, and of course maximally benefiting? So one might think, for example, about returning results to participants and how to do that outside of the usual frameworks that maybe we're used to in places like Europe and the US. How do we maximize the use of data? Some of that disproportionality in the number of participants in studies is because we have very large open access resources now in some parts of the world. These data get used a lot. How do we create resources to do that? And we've heard a bit about that today. But also, how do we think about going beyond this pilot study? How do we think about not just targeting one or two ethnic groups, one or two studies, but going much broader and really IHCC leading the way in addressing this ethnicity gap? Clearly, the resources required would be very substantial for whole genome sequencing, somewhat less if we were scaled back and just did genotyping and imputation. And I think that, of course, will depend on the appetite of scientists and funders to do something that's really innovative and transformative, or to do something that's more tried and tested. So I'll leave it there.